Welcome everyone. Drugs, which are substances used for the purpose of diagnosis, prevention, relief, or to cure diseases, could be derived from a variety of sources. So, in this session, we can have an idea regarding its sources. So, nature being the best source of drugs, we humans are also mimicking nature for our technology as well as for drug mechanism. Here, we are having the source of drugs which include both natural as well as man-made. First being plant source, and second one drugs obtained from animals. Third one, which are derived from minerals. Fourth one, microbial sources. Fifth one, that are synthesized from lab synthetic drugs. That last one, that is again a synthetic one that is produced from our lab by combining DNA fragments, recombinant DNA technology. So let us see one by one. So first being the plant sources, we know that our Mother Earth being carpeted with beautiful flora have been the source for number of valuable drugs, mainly because of its secondary metabolites. So what about the secondary metabolites? They are chemicals that are secreted by plants to protect themselves from predators. Okay. So these secondary metabolites are being analyzed for its medicinal values in particular. So here you can have a glimpse on some of the medicinal plants. This one, Rayulfia serpentina, from which an alkaloid, reserpin, is obtained and is used to treat high blood pressure. Apart from this, it can also help to cure mental disorders also. This is the plant with white flowers. And the next one, Digitalis purpurea, having pink bell-shaped flowers. And from this plant, glycosides, that is digoxin, deoxygenin, in these glycosides have been extracted out from this particular plant, and it is finding its application to treat cardiac disorders. And the next one is Vinca rosea. Two important alkaloids, vincristin, vinblastrin, have been extracted out from this pink flowered plant. And this acts as a natural anti-cancerous agent. And apart from this, here number of plant sources are given. Poppy, from which morphine, a painkiller, is obtained. Then cinchona, from which anti-malarial drug quinine is obtained. And to the bottom, daffodils, from which galantamine, which helps to treat Alzheimer's disease. Then digitalis lanata, from which digoxin, again finding its application as cardiovascular drug. And next one, white willow, being the source of aspirin. And last one, eucalyptus, from which eucalyptus oil having useful chemicals, this is pinene, cineol, limonene, terpineol has been used for its medicinal purpose. It has been popularly used as expectorant. And apart from this, number of commonly available plants are also here given. Aloe vera, neem, Philanthus samaras, then turmeric, ginger, garlic, then spices, black pepper, coriander, mint, then tulsi, uncountable variety of plants are available in our environment. And also, if we look at our kitchen items, number of spices, cumin, fenugreek, and coriander, such every items will have its own medicinal value. So it moved to explore out. So the next source being animal sources. Animals being the promising source for a number of valuable drugs. And these has been derived from mammals, birds, reptiles, and also from fishes. And the major active principle includes hormones, enzymes, and apart from this, vaccines, vitamins are also derived from this animal sources. And here you can have a glance on some of the drugs obtained from animals. From pigs, we are obtaining hormones like thyroxine, insulin, then anti-blood coagulant, heparin, then pancreatic enzymes, which helps for digestion. Then from horse, primarin, which helps to prevent osteoporosis and also used to treat prostate as well as breast cancer. Then from animal cartilage, chondroitin, that helps to treat osteoarthritis when it is combined with manganese ascorbate, glucosamine sulfate, glucosamine hydrochloride, and N-acetyl glucosamine. And also from animal bones we are obtaining calcium. Then from animal blood we are getting iron in number of vaccines. And then from shellfish, glucosamine, omega-3 fatty acids, calcium, etc. we are obtaining. Then cod liver oil. Then from sheep we are getting lanolin, a beautiful moisturizer. Then vitamin D3, which helps against bone loss. So all these drugs have been derived from animal sources and yet more to explore. And to our surprise, number of animal venoms have been analyzed to have its medicinal property. So let's explore some of them here. So animals that are secreting toxins to prevent themselves or protect themselves from predators, and these toxins are being used as medicinal products. The first one being nicotinamide, that is derived from cone snail venom, a seabed snail, and it is acting as a good painkiller compared to morphine. And the next chemical being tosuluristide. In this tosuluristide is obtained from scorpions, and it has been diagnosed to have benefits, that is, it helps to prevent brain damage, and it helps to diagnose brain tumor cells, and from which we could able to treat brain tumor. And the next one, the chai 1A, that is obtained from spider venom. It's a peptide and helps to control stroke by protecting our brain cells. And the next one is glumacin, that is derived from spider venom, and it helps to kill skin cancer cells. And last one, captopril, that is obtained from snake venom, helps to control blood pressure. So these are some of the drugs that has been explored out from animals. So the next source being minerals. So our Mother Earth being enriched with minerals, these minerals contribute for number of medicinal products including metals, non-metals, hydrocarbons, and also some clay materials also. And apart from its requirement for normal human functioning, They are the building blocks of life-saving medical devices also. For example, copper, titanium, which find its application in surgical instruments as well as MRI scanners. So here you can have a glance on some minerals that is acting as valuable drugs. Magnesium, zinc, gold, iron, mercury, bismuth, all these metal salts are having its own medicinal property. Magnesium salt is antacid, zinc salt to protect skin, and also it helps to extend our life. Then gold salts to treat arthritis, then iron salts to treat anemia, mercuric salts for syphilis, then bismuth salts to treat peptic ulcer. It's been derived from our minerals. And apart from this, non-metals like iodine is having antiseptic property, and hydrocarbon such as liquid paraffin is having purgative property that is being used as mild laxative for children's. And also kaolin, a type of clay, helps to treat diarrhea. So here you can have a glance over some of the compounds that from which we are getting medicinal values. It is all these drugs are being obtained from minerals. Okay. So next we shall see about microbial sources. So microorganisms such as bacteria, fungi, algae have been yielding large variety of antimicrobial agents. And apart from this, number of anti-cancer as well as anti-tumor compounds have also been recorded from different microbes. So let's explore on some of its types. So first one from bacteria. This is from Bacillus as well as actinomyces species. We are obtaining number of antibiotics. It is here you can see apart from this antibiotic obtained, there are anti-tumor compounds, then anti-inflammatory drugs, then anti-cancerous drugs, and immunosuppressive agents are also being obtained from bacteria. Okay, so here again list of it, and the next from algae. One of the product from blue-green algae, or otherwise known as cyanobacteria, that is spirulina. This product obtained from this blue-green algae is having its health benefits. And apart from this, it is acting as a good antiviral, anti-cancerous, anti-protozoal and immunomodulatory drug. You can have a view over it, some of the anti-cancerous compounds that has been derived from blue-green algae. So the next one is drugs obtained from fungi. Right from the age of Alexander Fleming, discoverer of penicillin, number of antibiotics have been obtained from fungi. And it will be acting as antibacterial and antifungal, then antiviral agents also. And apart from this, it is having anti-tumorous property also. So this is about microbial sources. And the next source is recombinant DNA technology. That is, in our lab, we are cloning two different DNA fragments, that is from different species. DNA from different species have been combined to form a new DNA fragment. Since the particular newly formed DNA fragment will have its own medicinal genomic property that is particularly applicable for deriving number of life-saving drugs. You can have a view on this YouTube clip. Here you can see a particular DNA, and this is the DNA fragment, and here this is an enzyme that is cleaving this particular DNA fragment, and by cleaving this particular DNA, a new DNA fragment is cloned with this DNA fragment with the help of enzymes. So, this newly formed DNA is helping to derive out number of drugs because of its peculiar genomic properties. This bacteria that is cultured from here is being used to derive out variety of medicines. Okay, so the next source being our synthetic drugs that is synthesized in our lab. So if we see this modern world, we today, that is drugs used by us and the drugs that are produced in our market mostly will be synthetic. And here you can have a glance on this video which explains about drug manufacturing.
Everyone in the Western world will have taken medicines at some time in their life to help combat a disease. Typically, for every drug that reaches the market, a pharmaceutical company, such as GlaxoSmithKline, will make 10,000 new chemicals. The vast majority will fail because they're not effective enough, or are toxic, or have unacceptable side effects, for example. Traditionally, organic chemicals are made, chemists use the word synthesized, one at a time in apparatus like this, which will produce about a gram or so of compound. Each stage of a synthesis can take up to several hours, and many stages may be needed to make the required substance. Modern techniques of screening chemicals for biological activity require only a few micrograms of compounds for initial testing and can be carried out rapidly with the aid of robots like these. This means that new techniques for synthetic organic chemistry are being used to make small quantities of many chemicals quickly. This technique, called combinatorial chemistry, allows many compounds of a similar type to be made simultaneously using a robot, a group of computer-controlled syringes. Normally, compounds are made in arrays of 96 on a plate with 12 rows of 8 wells. The search for a new medicine will start by identifying a disease that is not effectively treated by existing medicines. Chemists don't make compounds at random. The starting point for a new medicine might be a chemical that exists naturally in the body, or an existing drug, for example. Or scientists might understand something about the disease, that it involves an enzyme, for example, and speculate that a compound that interacts with the enzyme could affect the course of the disease. One approach is to synthesize large numbers of compounds with small variations in structure. These can be tested, screened, for possible activity as drugs by seeing how well they bind to an enzyme, for example. These tests can be done on isolated enzymes or cells. They're called in vitro, meaning in glass tests. chemists will develop a general reaction scheme that allows many similar compounds to be made by the same route. To take a simple example, 96 different compounds, called esters, could be made by reacting 8 alcohols with 12 carboxylic acids. Once this sequence has been programmed into the computer, the robot will add the appropriate reagents to the reaction vessels in the correct order. The reaction scheme can have any number of stages. In combinatorial chemistry, reactions are often carried out on beads of polystyrene resin. The starting material is bonded to the resin, and the new molecule is built up in stages. Finally, the product is broken off, cleaved from the resin using a strong acid. Compounds can go for chemical analysis and biological testing still in the 12x8 plates that are accepted by all the instruments. Promising compounds from this stage, called lead compounds, need to be made in larger quantities for more detailed testing. In this apparatus, developed at GlaxoSmithKline, larger quantities, around 10 to 100 milligrams, of the more promising compounds are made. This technique is called parallel synthesis. It can replicate all the traditional chemical techniques, such as stirring, refluxing, and so on, but is quicker because several compounds are still being made in parallel. Once a compound has been chosen for larger scale testing, normal ground glass apparatus like this can be used to make quantities of around 1 gram. A further scale-up to apparatus holding between 3 and 20 cubic decimeters can produce quantities of material of between 1 gram and 1 kilogram. The final stage before production is to make compounds on a pilot plant. This can make up to 100 kilograms for clinical tests on patients, and it can also be used to try out the techniques to be used on the manufacturing plant. Handling chemicals in these sorts of quantities is a far cry from robot syringes and even spatulas. By the time a product is being manufactured for sale, 10 years may have elapsed since the initial idea, and £450 million spent on the process.
Hope you find this session useful. Thank you.